Good evening, Miss. Thank you for the opportunity to present my part of our presentation. My part was pre-mRNA to mRNA, and let's start with during the transcription, the DNA strand is processed in the nucleus and copied onto an RNA strand. This happens by means of transcription factors. So transcription factors are basically the factors that enable or disable transcription. When you have transcription factors, you have this specific bubble over your DNA strand that you wish to duplicate or copy. So for these factors, you have your RNA polymerase. That's the enzyme that enables the transcription in the first place. And then you have your promoter. That's the site where the transcription is taking place. Your transcription factor will bind itself to the particular DNA sequence in question, and it can either activate the transcription process, repress duplications during the transcription process, stopping it, or bind a promoter to help or start to speed up the transcription. The RNA strand that's produced is known as pre-mRNA because it has not been processed into mRNA. Pre-mRNA, while being transcribed, is matching one nucleotide from the mRNA strand to one nucleotide from the template strand. These are three things that pre-mRNA must go through to become a complete RNA strand. So when you have your pre-mRNA strand, it's being transcribed. It's matching one nucleotide from the mRNA strand to one nucleotide from the template strand. That would be like your adenine matching with your uracil, because mRNA strands don't use thiacil, they use uracil. So it'd be your adenine strand matching with your uracil, your guanine strand matching with its reciprocal, and so forth. It's the same way how DNA would work. It's how your mRNA strand would match up with the template strand. So for you to have your data is processed and it's coded, for you to stop your processing, it has to have a terminator. The terminator is, as the name says, in charge of ending all the coding when it has all the proteins necessary to make the molecule needed. So when you have your terminator and you're stopping your transcription, there are three things that need to happen in order for your pre-mRNA molecule to evolve into a mature mRNA molecule. So when you have your pre-mRNA molecule, you have your normal strand with all of the parts that it needs, it doesn't need, and it's lacking some in. So first thing you're going to add is your 5 cap. That's at the 5 end of your DNA strand. You're going to add your 5 cap, and it's like a helmet of sorts. So, when you're little and your parents are like, you need to wear a helmet when you ride your bicycle in case you fall. That's basically what this does. It protects the strand, keeps it from getting broken down or disturbed or anything while it's on its way to make a new protein and cell. So it's basically the protector of the strand. It needs to be at the forefront at all times. And then at the 3 end of the tail, you have the addition of a poly A tail. Now, when you have your poly A tail, you need to have a polyadenylation signal. The polyadenylation signal allows the enzyme to know when the end of the chain is being cut off by the terminator, so you could add the poly A tail. Your poly A tail is a string of adenine molecules or adenine nucleotides at the end of the chain. Those are added by another enzyme. The addition of the tail stabilizes your new mature mRNA strand, but it's not quite finished yet. There's one more process that you need to go through, and that's called splicing. Splicing is the removal of your introns. Your introns are the parts of the cell, are the parts of the new strand that you don't need. They don't contain any genetic information that's useful. You don't need those parts. They're basically empty. And your exons are like your exterior parts, the parts that contain all of the important information that you need. So splicing is basically the removal of your introns. When you remove your introns, you have the 3 parts of your exon left that you will merge together to give you the full mature mRNA strand. As you can see in this picture here, you have your exons on the outside and you have your introns on the inside. When you splice, the introns are thrown out and you merge these 3 and you have your mature mRNA strand. So a little recap of what I said. You have the addition of a 5 cap to the beginning of the RNA. That's basically this little part right here. That's your 5 cap. The 5 cap also, I didn't mention, the 5 cap is made of a guanine nucleotide. Yeah, that's why there's a G there. It's a guanine nucleotide. And then you have at the end of it your poly A tail made of adenine nucleotides. So at your 5N, like 5 cap, so that's a way you could remember that. And at the opposite end, your 3N, you have the poly A tail. After you splice, this is your complete molecule. And you have the introns. You could call those your junk sequences. You don't really need that. You could call that the trash chute then. Like that's the trash of your laptop, so you're gonna get rid of it. So okay, that's the trash of this molecule, this DNA strand, so you're gonna get rid of it. You're only gonna keep the good things, like your good memories on your computer, your pictures with your friends, and you're gonna merge that all together and keep it. And then you have your mature mRNA molecule. Thank you for listening to my part of the presentation.